that is too small to be seen with our naked eye. That is, a microscope is required for viewing such organism. And in a much smaller case, as in the case of viruses, an electron microscope is required in order to view the organism. Microorganisms exist in different forms, and these include viruses, bacteria, protozoa such as amoeba, paramecium, euglena, chlamydomonas, certain fungi species, and of course certain species of algae. Microorganisms can be found almost everywhere. They are described as ubiquitous. That is, they can be found almost everywhere. They can be found in the air, in water, in the soil, in food, and in the bodies of other living things, and of course on their bodies. Examples of microorganisms found in air include certain viruses, such as the influenza virus and the common cold virus, the measles virus, and of course the COVID-19 coronavirus. Example of bacteria species found in the air includes those that cause respiratory tract infections, like those that cause pneumonia, that is pneumococcus species, the one that causes tuberculosis, that is Mycobacterium tuberculosis, and certain skin infection bacteria, that is Staphylococcus species. Fungi found in the air include fungal species that cause bread mold. Microorganisms found in water are of two categories. There are those that are naturally found in water, and there are those that are washed into water bodies from the soil or are introduced into the water bodies from sewage. Examples of those found naturally in water include the blue-green algae, diatoms, volvox, and other algae like Spirogyria. Bacteria species naturally found in water include Micrococcus, Azotobacter, and the Pseudomonas. Protozoans are also naturally found in water, including amoeba, paramecium, chlamydomonas, and so on. And some other primitive fungi are also found naturally in water. Those that are found in water due to the fact that they are washed down into the water from soil or from sewage. Those that are washed into water bodies from sewage are usually pathogenic in nature, and they include viruses like the hepatitis viruses, the polio virus, and so on. Bacteria from sewage source include the typhoid bacteria, Salmonella typhi, the cholera bacteria, Vibrio cholerae, and E. coli, Escherichia coli. These are known to be pathogenic in nature. Certain pathogenic protozoas are also washed into water bodies for sewage, and this includes the microorganism that causes dysentery, that is the Entamoeba histolytica. Soil microorganisms are known to break down organic matter in the soil. These are referred to as decomposers, and they also include certain species of bacteria that play a vital role in nutrient cycling, like those that help to fix nitrogen into the soil. Examples of these include Nitrosomonas, Nitrobacter, Azobacter, and nitrogen fixing bacteria. Also, certain fungi are found in the soil, where they also act as decomposers helping to accumulate organic matter in the soil. Food microorganisms are usually present in 3 different ways. They may be present as food spoilage agent, food poisoning agent, or food processing agent. For example, fungi and bacteria are notorious in the spoilage of food. That is why food items are kept in environments where these organisms cannot thrive, like in the freezers and the fridges. Those that cause food poisoning release certain toxic materials or substances called toxins into food substances, like the botulinum species that causes a food poisoning called botulism. And those that are present as food processors are deliberately added by humans, as in the case of bread making, cheese making, yogurt making, and fermentation processes in different industries. Microorganisms found in the body and on the bodies of other living things exist as what is described as normal body microflora. In fact, every healthy individual organism must have certain different species of microorganisms inhabiting its skin and its internal body surfaces. For example, these microorganisms found in the body can be found in the mouth, can be found on the skin, they can be found in the gut, and in females, some are found in the vaginal region. For example, those found in the skin include Staphylococcus aureus and Staphylococcus epidermis. Those found in the guts are known to help to digest certain carbohydrates that cannot be digested by the digestive enzymes of mammals. For example, in the digestion of cellulose. Some of these bacteria help to digest cellulose and release certain vital nutrients like vitamin K. Those present in the mouth, example Actinomyces viscosus, cause plague if the individual involved does not brush regularly. Another microorganism found in the body include certain species of fungi, particularly yeast. Sometimes it can be found coating the guts and even the tongue of a certain individual person. Growth of microorganisms. Growth of microorganisms or growing of microorganisms is also known as culturing. Microorganisms are grown or cultured in laboratory for different reasons. They may be grown in the laboratory to determine the cause of a particular disease. They may also be grown in laboratory to produce certain vital substances. The culturing of microorganisms or growing of microorganisms is done in special containers such as petri dishes containing a nutrient medium which supplies nutrients to the growing microorganism. A nutrient medium may be solid or liquid in nature, and the process of culturing involves a technique known as aseptic technique, which is a technique that makes sure or ensures that only one type of microorganism is grown at once and excluding all other microorganisms in the environment. Growth of microorganisms. When we talk about growth of microorganisms, we are trying to measure the increase in number of microorganisms in a liquid medium. So growth in microorganisms is expressed in the sense of increase in number of cells and not increase in the size of the cell, as the case may be in other living organisms. So in growing of microorganisms or studying the growth of microorganisms, number of cell or population of cell is taken into consideration. Most microorganisms are known to produce or reproduce by dividing through a process known as binary fission, whereby a single cell divides into two daughter cells, and each of these daughter cells divides after growing to its maximum size. A very fast dividing microorganism may divide every 20 minutes. Although when the growth of microorganisms is discussed theoretically, we shall refer to it the growth of microorganisms has been exponential in nature, although this is not the case in the practical sense, because growth of microorganisms in a closed system, that is a system containing certain amount of nutrients, is added into a liquid medium. So in short case, no extra medium is added, and at the end of the day, the number of cells that is obtained from the time of inoculation of the cell to the end of the study is determined and plotted on a graph. So such growth is not continuous because certain factors regulate or inhibit the growth of microorganisms in the liquid medium and its nutrient medium. Such factors include nutrient availability, space of the container, environmental factors such as water, radiation, oxygen supply, temperature, and pH. So in the growth of microorganisms, or the study of microorganisms growth, 4 stages are observed when such a particular microorganism is cultured in a closed system. These stages are the log phase, the exponential or log phase, the stationary phase, and the death phase. When the number of cells in such study is plotted against time, these 4 stages are observed. There is a section of this graph that shows the log phase, section showing the exponential phase, a section showing the stationary phase, and a section showing the decline or death phase. So these are measured by taking samples from the medium at time intervals. So the first stage observed, or the first phase, is the log phase. This is the phase during which the microorganism that is added to a new nutrient medium is taking time to get used to its new environment. That is, it's trying to adapt to its new environment, and then feeding. So at this stage, no cell division takes place. So during this stage, the microorganism adapt to its new environment. There is no increase in number of cells. Of course, the microorganism size will increase because it feeds, so there can be cellular weight gain because of feeding by the introduced microorganism. Immediately following this phase is the exponential or log phase. During this phase, the microorganisms introduced start dividing rapidly. That is, dividing and creating more cells. And each cell produced also divides because of the environment that is conducive for such activity. At this stage, microorganism keeps multiplying and is known as the phase of fastest growth, where most active growth of the microorganism under study takes place. When number of cells during this phase is plotted against time, a log straight line graph is obtained, and that's why it is referred to as a log phase. The stationary phase sets in at the time when the number of cells in the culture medium has increased to the maximum carrying capacity of the containing vessel. So at this stage, competition sets in for food and space, and most of the cells in the culture medium stop dividing. Following this stage is the death phase, when the cells in the nutrient medium start dying due to the depleted food nutrients and the accumulation of waste, thereby creating a kind of unconducive environment for the cells to survive. So at this stage, the cells start dying, and their death is also observed to be in an exponential manner or fashion. So when the number of cells at this stage is plotted against time, a straight line is obtained as shown in the graph earlier. Thank you for listening.